The issue of chemicals in the environment is a story about science, politics, health, and humanity's relationship with the planet. My name is Chris Hughes, independent scientist and founder of Embark, and this is the Chemical Journeys podcast. Join me as we uncover the story of chemicals in the environment through conversations with individuals working in this space. I hope to take you on a fascinating journey full of enlightening ideas, differing perspectives, and new understanding. So, let's get started. Hello everyone, and welcome to the Chemical Journeys podcast. My name is Chris Hughes, and I'm here today with Paul Thomas from Kreatis. Paul, thanks for joining me.
It's really a fantastic opportunity to be able to give us the QSAR side of things.
So, just a few words on why I've invited Paul. So, Paul and I have known each other for a few years. I think we first met back at the CTAC Basel conference when Paul was just starting out with his business with Kreatis. And I remember having a beer with you at your stand, which was really nice. And you've since had your own journey with building a business, which started around 10 years ago. So I thought it'd be really good to have you on here to talk about that, talk about your business and your experience. So yeah, let's get started. I thought we could start with, what is Kreatis? And give us a bit of the story behind that. How did it get going?
Okay, well, I'll give you a bit of a story about me. First of all, my background, I'm an ecotoxicologist. I was at University of Cardiff working under Dave Pascoe, doing my PhD, and then I came out to France actually on a grape picking mission while I was waiting for my viva at the end of the PhD, because I didn't want to be twiddling my thumbs waiting for that horrible moment. And I discovered that, contrarily to the UK situation, which was back in 1991, so years ago now, where the whole ecotoxicology market was basically saturated, back in France, they were just discovering the world of ecotoxicology. And the grape picking scenario was a great way to find out about it, because most was down in Bordeaux, and there were a lot of pesticides being used. And the first time I actually heard birds was after 6 weeks, when I started working in an organic grape farm, and that was a real eye-opener. It's the first time I'd seen a lizard as well for 6 weeks. So there was clearly something that needs to be done in France. So I thought, hey, this is an opportunity for a career. I came back out to France after I finished up with the PhD, and then moved into Paris, and fairly rapidly managed to get a job working as a study director and a manager of an ecotoxicology group, which I opened up within what at that time was called CIT, the Centre International de Toxicologie. All my companies, I think, more or less have changed names since I left. This is now Charles River in Évreux. I opened up their first ecotox lab, and that lasted for about 4 years. Fantastic experience, gave me a real understanding of GLP. And then after that, I moved to the chemical industry. That was ELF before it had been bought out by Total. Today it would be Arkema. Those guys were making some quite massive, well-known substances that I was working on, notably phthalates. That was a really interesting moment in my career, but also various other major chemicals, hydrogen peroxide, sodium perborate, and various others. Ultimately PFAS as well. So there were some really interesting opportunities to learn a lot about the science. And at that time, it was the risk assessment period 79393, the existing substances, which was like the precursor to REACH. So this was they really went into in depth, a lot of interesting discussions with regulators at that time and the rest of industry as well. And the thing I was missing at that stage was then a lab, and I got an opportunity to work with AkzoNobel, now called Nourion. And in that place, which was meant, I moved to Arnhem in the Netherlands. In that place, they had an ecotox lab, but there was also risk assessment going on, which at that time was environmental risk assessment. And I was made manager of a group. But that group then grew and grew. We soon took on a biocides lab, and then we took on a toxicology group, and then we took on regulatory affairs and safety data sheet writing. And before I knew it, I ended up with like 41 people working for me, and I was spending all my time doing management. And that was a little bit not really where I wanted to go. And I had good links with two guys from a consultancy in France called Cetra, so Philippe Adrian and Pierre-Gérard Pontal. Pierre-Gérard was a visionary toxicologist. Philippe is still working as an environmental scientist. And I had a good relationship with them, and I wanted to go back to France. I was kind of missing the mountains, not so many of those in the Netherlands. And it seemed like an opportunity to move towards REACH, which was just starting off. We were back in 2008 at this stage. So after brief interviews with those guys, I got to become the manager of the Lyon agency for that consultancy. I started in 2009, and I was using at that time some software. So it was the EpiWin EpiSuite software to try to make predictions, to try to help on the risk assessment side. I mean, I think we covered in the consultancy something like 700 different REACH dossiers over the period between 2008 and 2018. So there was just no time to be able to get experimental studies for everything. We were heavily reliant on alternatives. But EpiWin was kind of frustrating at the same time as it was an eye-opener. It was an eye-opener because it was so easy to use and so intuitive. And I really liked that side of it. On the other side, it was only right one time out of two, and you were never sure which time it was going to be right. So it was a bit of a difficult tool to be able to have a lot of confidence in. And I thought that I was basically a non-believer in QSARs up until about 2012. In 2012, I was asked to chair an ESITOC group, looking initially at critical body burdens, but actually that moved pretty quickly to chemical activity approach. So that was something that had come in from Don Mackay, a seminal article in 2009, which really, I think I could say was the beginning of Kreatis. So I worked with that group for a couple of years before we managed to produce something, and it was all based on kind of Verhaar methodology of splitting up modes of action and looking at the four groups and seeing if there was something that came out differently with the chemical activity approach. And it was pretty clear to us that something really big was happening if you use that approach. I came back at the end of that with a report in my hand, and I went to see Pierre-Gérard and I said, listen, Pierre-Gérard, we've really got to think about setting up a department looking at QSARs. I think it's going to really work. And he, being the visionary kind of guy, he said, "Oh, department? Well, why not set up a company and you'll run it?" So that was 2014. I founded Kreatis, and we were 100% R&D company. We were really very much a startup, and we were starting out with what we'd come out of ESITOC. But I had some ideas that Verhaar probably, as original as it was back in 1990s and year 2000, it probably was lacking some detail to be able to get a better acuity in terms of predictions. And this is where it all started to come into my mind, that ultimately we needed to look at a different methodology, perhaps looking at molecular initiating events, and try to categorize substances into more molecular initiating events, and then perhaps be able to link QSARs on the back of those. And that started off a PhD funded by the French authorities and by Kreatis, with a guy called Franklin Bauer, who's still working with us today. And his mandate was to try to create a scheme that was a little bit like Verhaar, in as much as it would contain a minimum number of classes that would cover, preferably not just for environment, but for human health as well, molecular initiating events that we could then use and structure into subclasses so that we could create QSARs on the back of those. So the important difference between that and the approach of other QSARs is that most QSARs are based, they're structural nuts. We're almost QMARs rather than QSARs. It's mechanistic activity relationships. And that is a big difference, because what it means is that you can have huge differences in structures that no read across would ever pick up. So for example, we have a MECOA 3.1, which has, amongst other things, aldehydes and epoxides in there. At least in ecotoxicology, we see that you can put those on the same regression line and they come out identical. And as long as you're using like moles per liter as your units, and we use it as log moles per liter, you can get very tight regression lines using both epoxides and aldehydes on exactly the same regression. And this is the real key of the mechanistic approach. It's about the fact that they have the same mechanism of toxic action. In this case, MECO 3.1 is hard electrophiles. And so the key event, the first key event, the first impact where the substance meets the tissue is identical for both epoxides and aldehydes. And so we can expect the downstream events and final parts of the AOP to be similar. And that certainly in ecotox comes out really clearly. We're now working on this area also on the human health side, and we're seeing very similar things, not for absolutely everything. I think there's like iron, skin irritation, or maybe acute toxicity that are somewhat different issues, but there are quite clearly very strong similarities we see within sensitization. And I believe that when we push this further to chronic QSARs on reprotoxicity, for example, there's no reason why MECOA shouldn't play a role within that. So that's really very much the motivations. We have some, just to finish off, we have a few extra concepts within Kreatis, which is perhaps somewhat original. We have some principles of what we call KISS, keep it simple, stupid. So we don't start off with pharmaceuticals. You know, I mean, those things are really, really difficult to be able to determine in terms of mechanisms and also downstream in terms of QSARs. So you start off with like non-polar narcotics and then gradually you work up. And now we've got like 28 different mechanisms of toxic action. Not all of them are today linked to QSARs, but many of them are, and we're just adding as we go on. We want to base on foundation principles. So again, we'll start with those simple ones, and once we've got the simple ones sorted out, then we add to that, and we can discern levels of complexity that we had no idea. 9 years ago, we just had a couple of cases. This anilines is a nice one. We just I think worked out what the issues are with anilines. Anilines are incredibly complex in terms of their toxicity, and it's about hindrance. And it's taken us 9 years to work that out. So it's all about being based on mechanisms and not just structures from our side. It's also very much about the validation of studies. So every single study that we see, doesn't matter what it is, we do our best to actually internally validate it. So in terms of the staff that we've taken on chemists, we've taken on human health toxicologists, environmental scientists, that they're all postdocs. They all have a really good understanding of the science before they even join us. And then we spend quite a lot of time training them in the techniques that we use internally. And so the first thing is, it's incredibly time consuming, but the first thing is, before we take a study and do anything with it, we actually check that it really is valid. And it's quite shocking if you look in the ECHA disseminated database, just how many studies actually are not fit for purpose, even though they're scored Clemish 1 or Clemish 2. They are, about on average, I think we're finding 1 study out of 2 is not strictly valid. And sometimes as well, some of the things which are noted Clemish 3 or Clemish 4 actually are valid. So that you can't even count on the invalid ones as being invalid. We don't use any studies before we've validated them, although that's getting, of course, more and more complex when you're looking at thousands of studies, and we're setting up a biodegradation QSAR now. That we're using literally thousands of data for that, but we still end up going on a case-by-case basis. We're spending days going back, looking at robust summaries, looking at reports, trying to check whether these studies make sense or not. So it is a real founding principle of Kreatis. The other things are, we want to be sure that we're following OECD and ECHA principles for QSAR production, of course, so that's really important for us. We're trying to produce studies that are predictions which are as good as experimental studies, and maybe even, and if I dare say this, sometimes better. Not just better because 50% of them are invalid, but better because I think if you're lining up a whole bunch of different data on similar things, you actually might be able to get a better discernment than a single empirical study. So I think that is often not recognized, and QSARs are seen as kind of there, and I can understand why, given the backgrounds. QSARs may be seen as a kind of approximation, but actually I think QSARs can give you far better insight. I think I've ever learned as much about ecotoxicology since the last 10 years that I've been working in Kreatis. That's really been a step change for me in terms of my understanding of the science. And another thing is we're trying to push for good modeling practice. So we seem to be a little bit out on the periphery on that one. It seems there's not an awful lot of supporters for good modeling practice. But as I see it, if you have labs which are forced to do good laboratory practice, I don't see why us as producers of QSARs have got to get off scot-free and not have to make any effort to demonstrate that we're producing quality material. And the last thing, of course, is we're struggling now to see the best way to include artificial intelligence. Everybody's talking about artificial intelligence. The question is, how are we going to use artificial intelligence in a naturally intelligent way to make our models better? Because you can also make total rubbish with artificial intelligence.
Yeah, we've all played with ChatGPT.
Yeah. Look up your own profile, and you'll probably find total falsitudes in there.
No, thanks very much, Paul. That's really a really nice introduction to your journey, and yeah, fascinating to hear about the journey that you had before you started Kreatis. That was really rich in experiences and insights working across different sectors. So I'm intrigued just to kind of, if we just reverse back to that point where you were looking to start out this company, and perhaps you could take yourself back to where you were there and reflect on, how are you feeling at that time, and what were the kind of key things that drove you to doing this? I know you mentioned you had this mentor, visionary scientist. Sorry, I didn't catch his name, but were there other things that drove you towards that?
Yeah, I think it was Don Mackay was the sort of demigod of environmental science. It's a guy I've been talking about for many decades. Before that, chemical activity, phase equilibrium thermodynamics. It's related directly to fugacity, but specifically in our case, in terms of water. But it can also be, at least according to his seminal article in 2009. It can also be related to things like inhalation toxicity for rats. So it just depends what medium you want to put your substance in.
Yes.
And it really started off with that work. I think I had to read that publication at least 6 times before I understood it. And then when we started working on it, and initially already within the ESITOC group, we started looking at validation of data. So we already threw out within the group a huge number of studies, but then we misclassified a load of stuff. I think we ended up putting phthalates in their HAR1 class as non-polar narcotics. So there were clearly loads of errors in there. And then so when I took that back to the consultancy afterwards, and we started working on it internally, and then the other thing which we realized was that we needed physchem as descriptors. So this then became a major subject. Most people, when they're doing QSARs, in ecotox they'll use log P, log KW as a descriptor. The chemical activity approach considers that there's a direct relationship between toxicity and water solubility, and not log P. And in fact, we realized after a fairly short time that you can have loads of different relationships between log P and solubility. That's very much structural group dependent. So there's not one direct, Ran et al. 2002, I think, came up with a general solubility equation, but that is a very general equation. It's not specific. The bad news for us was that water solubility is just one of those endpoints that no one really cares about. So they're not very well checked up on in labs, and there are enormous differences in the methodologies are not recommended in OECD 105. You can either finish up with emulsions in your shake flask, which if they're stable emulsions, then you're stuck with it maybe for 24 hours. So you can measure massive amounts of substance in there thinking it's dissolved and it isn't. And if you use the column elution method, some people even use that with liquid. So it's completely invalid. But even if you use it with solids, because of the flow through rate, if the water solubility kinetics is a bit on the low side, you're never going to reach saturation. So you're underestimating water solubility in those cases. So the two main methods which are used are basically inappropriate for many chemicals. And we switched to using an OECD 123 slow stir methodology for log P to apply that to the water solubility, and often worked quite well. And then we were finding we were getting much more appropriate water solubility data. So there was a bit of quality water solubility data out there in REACH, but in fact, the best data was coming from log P, and even that had some issues with it, but most of the quality data was log P. So we had to go through, what we do with our platform is we start off with a structure, and then from the structure we go to the log P, and then from the log P we determine water solubility, and then from the water solubility we look at the MECOA, and then or mechanism of toxic action, and then from the MECOA we go to the aquatic toxicity QSARs, and now we're starting to do it a bit with the human health ones as well. And we realized that we needed to create these descriptor QSARs because you couldn't always trust the experimental data on water solubility or log P. So you can't always trust the QSARs on water solubility or log P either, because of course QSARs quality is related to data quality. So this is another area which we're working on. And now in a future version of the model, we've got a new AI-based model which has a much bigger applicability domain than our classical QSAR model. We're going to introduce that within our platform beginning of next year.
Oh, that sounds really exciting. Yeah, I'd love to talk about that a bit more with you. And what you're saying was resonating with me because, in my past, I've done quite a bit of work on water solubility myself. I'm aware that you can get these huge inaccuracies in experimental measurements, especially when you're working at very low substances with low water solubility. And I was actually involved in drafting a proposal for a Cefic LRI project, which recently concluded, where they developed the protocol for measuring water solubility with greater accuracy. I'm trying to pull it away from the technical for a moment and look at the situation. I mean, was this something that, did you have to give up certain things, when you were starting out on this journey, or did you take, were there any risks involved, or were you more kind of, was it clear to you that this was the avenue to go down? And how did you deal with that uncertainty in the early stages in terms of, getting your first contract or, sort of starting off in the business?
Yeah, well, as I mentioned, I was managing this group in Lyon. We were doing pretty much exclusively REACH work with that group. And then we'd captured a lot of the fragrance industry within the company. So we were doing a lot of risk assessments for fragrances. They had a huge number of molecules, and there was a big need to limit the amount of testing for these substances because there were so many of them, and to try to find a better way of testing them. And so we kind of moved Kreatis and the fragrancy industry together so that we could make better predictions for the typical kind of molecules that were in the fragrance industry. And the good news, I mean, the fragrance industry, they're not producing like pesticides, they're not producing pharmaceuticals. So you're in some of the easier mechanisms of toxic action within those cases. They're not always that easy, but many of them were. And there was a lot of data available, basic data, which was available to allow us to build the QSARs for it. So that helped. It was a useful combination to have on the sidelines of the consultancy an in silico group that was able to instantly react to requests coming from the consultants. So I saw a real synergy by having these two companies together. But Pierre went off into retirement, and the company was bought by an investor, and I was made managing director of the company at that time. So Kreatis became a sideline occupation for me. I only had 15% of my time to work on it. You can't actually build a startup, run a business on 15% of your time. And the investor who bought it wasn't that excited about research and development. So I negotiated at that point to buy the Kreatis from him, and I did that with a colleague who was a leading toxicologist within my group called Carol Charmeau, and so we bought the company together. 50-50. And Cetra then no longer had any shareholding within Kreatis. And so we were kind of, after a partnership agreement, we were kind of, the umbilical cord had been cut. And it was a scary time. This was back in mid 2020. It was a moment of great uncertainty because 2018 for Kreatis had been a fantastic year, the best year without a doubt. Everybody was finishing up their reach dossiers. They desperately needed data. They were looking at opportunities to be able to get a reasonably cheap and fast result. So Kreatis became an instant star in 2018. But in 2019, everyone had handed in their reach dossiers, didn't need it anymore. And I think industry was taking a step back and thinking many of the business units, I think, believed that reach was finished. And
reach was done. Yeah, everybody took a breath and slowly made their teams moved on to different things, right?
Exactly, and
Their
it was like, no, we're
expertise
moving your team
drifted
to
away.
something else. No, you've reached is finished now. So there was not a peep out of industry for a year, and we had our worst year ever. I mean, we really took a hit, and luckily 2018 had been a good year for us because the losses we made in 2019 basically soaked up all the profits we made in 2018, and we held on to the staff during that time, but. Obviously, the investor wasn't too comfortable with that, so that was a good starting point for discussion. But I had doubts that we were going to make a success of this operation. At the same time, I realized that something in me had changed, so that in fact I'd become totally passionate about what we were doing, and I really believed 100% in it and in the future of it. And I just thought, well, if we can hold on long enough, maybe everybody else is also going to realize that there's a real potential future in the area of in silico. And at that time, that wasn't so much the case. Back in 2020, there was a huge issue around whether QSARs would be accepted even by ECHA. We'd had an exceptionally good relationship with the computational unit for many years. We'd always tried to be upfront with them about the data we were using, how we worked. We would try to have meetings with them every single year from about 2015 onwards, I think, just so that we could explain to them what technical progress we were making and try to get them to believe in us. They are a really good bunch of scientists, so they were really interested in the work, and I think they helped to motivate us every time we met them. I always came out with a feeling of, yes, this is going somewhere. But of course, ECHA was never officially pronouncing that they would accept QSARs. And it was always, yeah, well, we'll see, and maybe there'll be a good supporting study, or well, we'll have to see. It depends on which QSAR it is. And there were so many different stories coming out. You never knew which way it was going. And then something miraculous happened just this year on our 10th birthday. So we're delighted to hear this. What happened was CTAC Seville. And Doris Herman gave a presentation where she announced that ECHA would accept a QSAR as a standalone result in a reach dossier, obviously, as long as it met with the appropriate principles. Also, there was another thing that came out, was the QAF had just come out at that stage. I mean, we had spent an unpleasant end of 2023 completely updating our QPRFs and QMRFs, which I'm actually now, in retrospect, I'm really glad that happened, because it was a horrible job for us to update them, and it took us months. But I realized that at the end of it, that we'd really needed to do that to be able to demonstrate the veracity of the predictions that we were making. And I think our QPRFs now are much higher quality than they were before that. So I'm really glad that happened.
Yeah, that's great, and good housekeeping often does not appear to have a payoff while you're doing it, but then you look afterwards and you think, oh, I'm so glad that I had the discipline to go and do those things, or pay that cost. It's paying back now. Just to jump in there, you mentioned a few acronyms, so we'll just go through. We've got the QSAR, the QAF, the QSAR Assessment Framework, and then the QMRF, the QSAR model reporting format, and the QPRF, QSAR prediction reporting formats.
Yes.
Why don't you take us through a little bit of what those acronyms are about?
Well, yeah, I mean, in a nutshell, they demonstrate your applicability domain of your model. Applicability means something that we're always talking about in silico, but actually I think it also applies to every kind of study. Every study has its applicability domain, but QSARs, of course, they need to demonstrate they're within a certain set of applicability domains. So you can have a descriptor domain, so it'll demonstrate that if you're using water solubility, that your data fits within the maximum and minimum water solubility values that you have to demonstrate structural domain. So then you're saying, well, yes, okay, we actually have within our training set the same specific structures that we are, which of as our as the molecule that we're looking at, a molecule of interest. Obviously, if you have totally different structures, then you can't possibly consider that they fall within your applicability domain, and your prediction is going to be accurate. On the other hand, there are also, just going into a little bit more detail, certain caveats to that statement. The critical structural groups are those which actually could impact on the mechanism of action. If it's only a question of hydrophobicity, for example, because your alkyl chain is a little bit longer or a little bit shorter than the ones that you've got in your training set, that's not necessarily going to have an impact that you can't calculate. So that in itself wouldn't necessarily be a problem. So it may not fit strictly within your structural domain, but as long as you're not finding that, there's an aldehyde in there that I don't have in my structural domain, so I'm going to count it as a non-polar narcotic, that would be a major mistake. So it depends a little bit on what you're doing. Then you've got mechanistic domain, of course. So for us, this is the big one, even though within the OECD principles, mechanism is a nice to have. Unfortunately for us, it's like nearly a must. Obviously, within the MECOA, the whole principle of it is that the structure fits within a mechanism of action. So there's a mechanistic domain, and so we need to demonstrate that we're within a mechanistic domain. And then you can have other things like metabolic domain, for example. So that there are a bunch of things that you need to demonstrate, and all those have to be detailed within your, first of all, within your model reporting format to say what your model actually contains. And then when you're looking at the prediction reporting format, then you're specifically looking at your target substance, and does that target substance have similar training set data within the QSAR that you're looking at.
Mm-hmm, mm-hmm. Yeah, and all this is sort of working towards the documentation of your predictions so that you can establish confidence in them to ultimately rely on them for some element of a regulatory assessment. And I guess it's important to say that a lot of the work that you're doing in the in silico space is as a substitute for some experimental study, which can obviously cost an awful lot of time and resource and money to deliver those sorts of data. And as you've already mentioned, there are also some limitations to doing these experimental studies as well. So by and large, this is a good thing for QSARs that with the QSAR assessment format, we have now formalized or made more concrete this process for documenting and justifying QSARs. So how where are you on this? Has this been a major breakthrough? Is this merely documenting best practice that was already in place? Do you think the QAF is going to be a help overall, or is it going to make your work a lot more challenging?
Yeah, the interesting thing with the QAF is it gives you a checklist, and it's a checklist which was originally made. It's a little bit like the RAF, the Read Across Assessment Framework, where it's actually made for regulators, but it then gives you insight as a producer of the things that the regulators are going to look at, and of course they will make sense. And of course the statistics, we haven't gone into that, and I'm not a statistician. We have people who do that for me, thank goodness, but also has to be demonstrated as being reasonably good. And from that, you can then get an overall indication of the uncertainty of your prediction. It's very rare to find something where you're 100% certain that your prediction is going to be accurate. But then I say, well, okay, we could condemn in silico for that, and we should perhaps only say, oh, we can only take things which are 100% certain. But actually, if you look at experimental studies of any kind, how much certainty do you have in those? It's the same thing. You're always going to have some degree of uncertainty, and it's very rare that you're going to be 100% sure of the accuracy of your prediction.
Yeah, yeah. And I guess there's a good thing with this QSAR Assessment Framework in that it's an international effort, right? So ECHA's obviously
OECD.
been heavily involved
Yeah.
in that work, but it's an OECD framework. Because I've heard that, in particular with the read across assessment framework, that this has made it quite challenging to get read across accepted. It's sort of increased the burden of effort required, and often there's a bar that's difficult to meet in justifying read across. Do you see similar challenges with on the QSAR side, or are you more
It's
optimistic?
definitely increased the workload. I mean, we used to be we semi automated our QPRFs about maybe around the beginning of 2023, and we abandoned automation after QAF came out because we realized that it's very time consuming in terms of expertise, and it would be a nice to have that within our modeling platform that we could automate the production of QPRF, and there are certainly some platforms that do that kind of thing. But actually, when you look at the 9 sections which are required to fill in a QPRF these days, and now they've just brought out an update for the QAF. So when you're looking at multi-constituent substances, that's not, we're using it for that, but actually it's when you're looking at multiple predictions coming from different methodologies, there's a kind of consensus QPRF which comes out at the end of that. So that's yet another extra layer to go on top of the previous layers. So it means at the moment I'm quite happy with that, because what I see is that we have our modeling software that we sell as licenses to our clients, and we try to update that like once every 2 years, but twice a year. But in between times, in the 6 months before we bring out the new version, we've made 6 months of progress, and 6 months of progress in the in silico field is actually sometimes quite a lot. So what the client is using by the end of the 6 months period, we're sometimes way ahead of that, and we can see perhaps different things from what the client would see by using our software from 6 months beforehand. So I like it to some extent. I like the fact that every single one of our QSARs, before it goes to a regulator, is always validated internally by our own experts, so we can be sure. I mean, we have nothing to gain by sending a bad QSAR to the regulators. We have everything to lose. Our credibility is what counts for everything. And if credibility comes in walking and it goes out running. So if we start sending nonsense to regulators, or we start producing QPRFs which are inferior because we want to automate them and make things quicker, we will just end up losing more than we gain.
Yeah, you're definitely right there that credibility is very hard won and easily lost. So it's great to hear that is obviously coming through here. I suppose it is a way of kind of rewarding you for all the effort you've put into building this business so far, that it makes it more challenging for another organization that's maybe not being quite as diligent to kind of come in and start trying to compete with you. I guess something worth listing, perhaps, the different endpoints that you're able to predict for, if we could just do that quickly.
Yeah. So, well, as I said, we've split into chemistry and ecotoxicity at the moment, and human health. Within the human health, we also have an ED section as well.
ED is
endocrine disruption. So we have over 40 acute and chronic on the ecotox side. And we'll go for the classical ones, because what we're trying to do is to replace OECD studies. It's a big issue involved now in a lot of debate about NAMs, where new approach methodologies, or some people call it non-animal methods, which is debatable. It's more of for us, it's a new approach methodology. Yeah, and so we have the on the physicochemistry side, we have the log P and the water solubility. So that's it. And we have two major QSARs which are now being used on the log P side to try to get first an increased applicability domain, and also to get increased acuity. And then on the ecotox side, we have algae, daphnia, and fish. So as I mentioned, we're trying to use replacement endpoints. So this is what we are aiming for. If you're doing an acute fish study, then the QSAR prediction is a replacement for the OECD 203, potentially the OECD 236 if you want as well. The chronic study would then be a replacement for the OECD, the fish early life stage study. And given that the authorities have abandoned pretty much the other chronics or pseudo-chronic fish studies, we've really concentrated on the 210. So all our data are based on 210 and not anything else. And so this is, in some of the fora we go to, they mention that it is the problem with NAMs is that NAMs do not manage to accurately replace studies. But actually, the people who are saying that are talking about in vitro NAMs. They're not talking about in silico. And the whole idea of the in silico NAM is we replace the study. All of the data we are using are based on the data from that specific study. So if we're doing a chronic fish, it's based only on exclusively on OECD 210. So when we do a prediction, we're replacing the OECD 210. If you're going to use RTGW1, the gill cell, then you're not replacing a whole fish. So in that case, you could argue, well, oh yeah, okay, maybe it doesn't replace a whole fish, and there are issues around, okay, well, how do you determine for neurotoxics, etc. The idea with in silico is that we replace the study. And I think there's a common misconception, probably mostly coming from the in vitro crowd, where effectively you can't replace the whole study, but that doesn't make the in vitro null and void either. There is a place for both, and I believe that there is a potential for NAMs in silico and in vitro to be used in a combi approach when there is a lack of certainty on one side or the other, so that the in vitro approach can kind of validate the in silico outcome to demonstrate we've not missed an appropriate mechanism of action. Maybe we've not spotted something, because the problem with in silico is we can only know what we know. We can't invent mechanisms that weren't there. Whereas if you do an in vitro study and there is some insidious mechanism of action we're not sure about, then the study should pick that up in theory. So as a combi, you could use the in vitro to validate the in silico approach, and the in silico approach could mechanistically explain the results for the in vitro and provide a prediction result for the outcome on the in vivo study. So I think there's a potential for them to be used together, which would help them to become even stronger than current in vivo studies and current empirical methods. So going back to what we produce. So we follow OECD guideline type studies, and we're looking at OECD 201, which is the algae, and we do acute and chronic. We do the OECD 202 and 211, which is the acute Daphnia study and the chronic Daphnia study, and we do the OECD 203 or possibly 236 if you like, and the 210, which is the acute fish and chronic fish study. We also have the ASRIT study, which is the OECD 209, the activated sludge respiration inhibition test. I think it's the only one out there which does that. We're now looking at finishing up on our biodegradation model, so that should be coming out early next year, really early next year. We've nearly finished it. We have a pilot study on bioconcentration factor, which is looking only at currently at worst case non-metabolizable substances, and we're going to start working on that again as soon as we finished, I think, with the biodegradation. So the idea really is to have PBT, persistent bioaccumulable and toxic endpoints, covered, working towards the SSBD, so safe and sustainable by design approaches which are proposed by the Joint Research Centre, JRC, at this stage, and are very likely sooner or later going to come into force and presumably replace REACH. And under that framework, part of the Green Deal, the idea would be to have non-toxic substances, so they say, and there are limits to what you could define non-toxic as meaning, but assuming less than the classification value for toxicity, then it would be a useful indicator to have an in silico approach for this assessment method, because you need to look at chronic toxicity, not just at short term. If you're going to compare the potential of your substance, which might start off at 10 tons per year and have some acute endpoints on it, so it doesn't look toxic at all. Let's take DEHP as a fantastic example, diethyl hexyl phthalate, which became a multi-million ton per year substance. You look at the acute endpoints, it's totally harmless. You start looking at some of the chronic ones, and notably the endocrine ones and the human reprotox ones. And even on the fish, if you do a really long chronic study on it, you can start to see intersex. It is a bit of a problem chemical when you get to a certain tonnage. Now, the stuff is everywhere. And even though it's been banned in Europe for the last decade, it's still creeping in through imports, presumably mostly. So if we're going to look at safe and sustainable substances by design, then we need to be able to ascertain the chronic effects, not just the acute ones, in order to be able to decide what is safe. Because ultimately, those are the small substances today, which could be the big substances tomorrow. And you're not going to be saying, oh, well, hey, this is great. I found this fantastic substitute. And then it turns out to be more toxic than the substance you are substituting for. So the
Yes.
same approach. So
Yeah, we should.
Yep.
Yeah.
So
go ahead.
I'll just finish off on the human health. On the human health, we have some local QSARs. So eye and skin irritation. We have a really nice one on skin sensitization, which we're really proud of. And that one uses the mechanism of action concept, which works really well, but in combination with other concepts such as the impact of autooxidation and skin absorption. We also have a skin absorption model as well, and we're going to start looking at longer term effects as from next year. I think it's going to take us a while to get there, looking at repeat dose and reprotoxicity, but it's because, as I mentioned with the SSBD, it is really this is our mission really to try to move to these chronic endpoints for human health, which currently are really lacking. And there's the endocrine disruption one. So we were funded by an NC3Rs project by Syngenta and Shell a couple of years ago now, and we're just finishing up for the thyroid, both a thyroid docking model and a thyroid QSAR model, looking at endocrine disruption there. And that is really, I think we've made really big progress on we had some existing models, but they needed a huge amount of extra work to get them to be really top quality functional. I'm really proud of those models now. I think they're going to be great. They should be packaged by the end of this year or beginning of next year. And we also have the other E, A, and S models, so estrogenic, androgenic, and steroidogenesis models as well. But we're going to use the same validation procedure that we used for the thyroid to upgrade those models as well in the future. So I'm hoping we'll have a bunch of EAT and S endocrine models coming out next year.
Very good. I mean, it sounds like you've got quite the suite of different tools and models within your repertoire. I just want to go back to some of the things you raised there, because you brought up the issue of NAMs, the new approach methodologies, and also SSBD, safe and sustainable by design. I suppose on NAMs, you're right. There are two schools brought together, right? There's the in silico group, and then there's the in vitro group. And when people are talking about NAMs, there's been a huge amount of debate going on recently about NAMs and the moral and ethical need to reduce the amount of testing that we do on animals. And there's been this clash with the sort of legal requirements of our regulations like REACH, which require certain specific tests, which of course the in vitro NAMs do not match up to one-to-one. And so therefore there's this legal and scientific headache where regulators are not in a position to allow in vitro methods to be used in place of some of these standardized tests, but you have this. People have recognized broadly that in silico models and tools are very much part of this toolkit. But I think you're right to recognize that what people don't realize is that an in silico tool is often much closer to one of these standard guideline tests, and so may provide that bridge to help to enable these endpoints to be ultimately replaced, these tests to no longer be used.
Yeah, and I think it's simply that in silico is perhaps still a smaller field today. I think I'm an unusual case because I've gone from lab in vivo studies and then jumped to in silico replacement. And as I said earlier on, I was probably one of the greatest, non-believers in silico approaches, as seeing it as something which was a kind of quick and dirty approach compared to an actual experimental study that I suspect that many of the people coming from the in vitro world were perhaps originally in vivo and moved from one laboratory practice to another laboratory practice for a good cause. But it means that I think they often have little belief in silico, and it seems that it never the twain shall meet. The people who are heavily involved in vitro, they don't believe or want to believe in silico methods. And unfortunately, there's a kind of, I won't call it a war exactly, but there's maybe a competitive nature going on between these two forces. Whereas I think we could be, as I said earlier on, I think we could make a very good combination together, be more powerful, and certainly have more strength in replacing the current in vivo paradigm if we spend a bit more time collaborating. I go to these meetings on phasing out animal testing. In fact, the last time I went was in Brussels a few weeks ago, and the entire discussion was about in vitro, almost anyway. And I just brought up the point, there was a big discussion about validation, and I just brought up the point right at the end of the meeting that it seemed very strange that we were talking exclusively about in vitro, whereas in fact there are also discussions and debates to be had on the in silico. One of those debates, I mean, from my perspective, of course, as a commercial software producer as well, there seems to be also an intense debate about whether all QSARs should be freeware or not. And we're not the only commercial producer out there. There are others as well. And frankly, I think the commercial models seem to have great value, perhaps more so than many of the freeware models. And yet there seems to be this debate that, oh, well, if you're going to be able to use a QSAR, then it should be published, or all the data in it should be published. Well, obviously, if we do that, then we have no competitive advantage anymore. People could just take our software and use it for free, and we would go out of business. So it seems to be a very kind of complaint to make that for some reason we shouldn't be able to believe in QSAR manufacturers' production simply because they're selling black boxes and we don't know what's in them. Now, I'd see a very simple solution to this. It's in laboratories you have GLP. There've been descriptions of good modeling practice, what I would call GMOP. So if labs have to have GLP, why shouldn't QSAR producers have to have GMOP? We could very easily set it up. There are accreditation organizations in the UK, for example, it's called BARCA. In France, it's called COFRAC. And every country has its own authority for doing that. So basically, you're audited for a day or two by a specific third-party auditor who has no interest whatsoever in stealing your business. He comes and looks, checks at your lab, makes sure you're following your standard operating procedures, that he looks at 2 or 3 different studies that you've carried out over the last year to check that there's no cheating going on, that all the figures make sense. How difficult would this be to impose on in silico QSAR producers to say, okay, you can either be accredited or you can be not accredited. If you're accredited, then we believe in your software because you have everything to lose if you're caught, trying to cheat. So it's not a question of black box anymore. It would be so easy to set this up. And I mention it at every available opportunity. And it's kind of one of those things where it's okay, so that's fine. Now we move on to the next point. And it's nobody wants to take this on board. And yet I think it would be so simple to set up. If it works now for GLP and GLP labs, why wouldn't it work for QSAR producers? We are an in silico CRO.
Yeah, I mean, it's a really interesting point that you make. And, this is the thing about balancing your commercial interests and the investment that you have made with the desire in the scientific community for there to be transparency. And it's a real challenge. And I think you're right in that there does appear to be that sort of an institutional gap in having an organization that can check and validate and accredit the work of organizations that are working on QSARs. If you're happy to work with an organization like that, there's still presumably the risk that there could be leaks from within that organization that could still release your trade secrets or lose them to others. But the fact that you're still willing to accept that risk in the drive to have this issue solved, I think speaks volumes. And I agree that there is a need to promote the use of QSARs, and QSARs that emerge from academia, they often emerge sort of organically, or they will be balanced with other things, and there'll be other incentives that will be drawing people's attention towards different things. It's a different matter when you've got a commercial private organization that has made developing QSARs and bringing QSARs to serve a particular issue. It's a different matter ultimately, and you'll get a different outcome. Both likely belong in this ecosystem that we have. But yeah, I hope that you can make some progress with that push that you're making towards getting some independent accreditation.
Well, we keep trying, and of course, I mean, it's not really helped by groups such as PARC. Now, PARC is an international form of PARC, which is mostly run by academics, and are doing a great job. Come up with a nice piece of software now where you can go through a flowchart and methodology for looking at your substance, but it's all based on freeware. There is no room there even to suggest integration of commercial software, and yet nobody would say, well, of course, this is a tier one. So if you go to tier two, well, you can still do the in vitro study, or you can ultimately do the in vivo study. Or, okay, so what about a tier two where you just very simply can plug into some commercial software and you can use that instead of the freeware, which may be giving you totally unreliable results. But there is there's a need for a recognition at a much higher level to be able to go all through the business of in silico. And the other issue is, in silico is making, can naturally make much faster progress than in vitro can, because of all the hurdles that in vitro has to go through from a regulatory standpoint in order to get recognition in an OECD guideline. In terms of the number of inter-laboratory tests that need to be carried out and the creation of the guideline itself, these are all incredibly long-winded processes. So if we want to set up SSBD within a reasonable time frame, 5 years or something, many of those in vitro endpoints are not going to be finished and ready. Whereas I could see the rate things are going at the moment in silico, we could possibly have an entire framework set up within that period.
Yeah, so there's so much ground to be gained on this, right? And you're definitely right that those processes for validating the experimental methods are also very slow and cumbersome, and probably not well adapted to this modern day of risk assessment, where we're having lots of little batteries of tests that are designed to provide the same information as one big sledgehammer of one of the traditional OECD tests. Yeah, I think the acronym probably drives you mad is FAIR, isn't it? Freely available, accessible, interoperable, and what's the R?
I can never remember it either.
But that's the real buzzword, one of the buzzwords of the day, isn't it?
buzzword, but actually, and I've talked to people like Mark Cronin about FAIR, and he is a strong protagonist when it comes to use of FAIR, and rightly so. But he says to me, "Don't ever say that commercial software is not applicable. It's about use after production. So if your prediction is bought by industry and used once, then it's about actually the downstream use of that prediction to say at least, and in fact, even FAIR principles don't even say, well, now you have to give it away for free. It means freely accessed, but that doesn't make it free. You have to have some kind of mechanism where you say this endpoint prediction exists already. You don't need to generate it again. That's all FAIR is really saying. And it's not saying it has to be free from the beginning. And I think, again, there are often misconceptions about what FAIR really means. And a lot of people are taking FAIR to mean freeware.
Mm-hmm, mm-hmm. Yeah, and there's definitely a noble pursuit in making information more widely available, more transparent, and democratizing. I know that I've cursed certain software that's behind paywalls before, and I've also cursed the fact that a lot of scientific journals have been behind paywalls for a long time, but there seems to be some positive moves to make this kind of information more available. So I am very much kind of on the fence, and I'm not sure which is the right way to go. I would like to talk to you about your biodegradation QSAR if you're able to, or how much could you say about that? And I think the thing specifically is that biodegradation, environmental persistence is an area that I'm really interested in. And one of the things that has been a major issue with these properties is that the models that are available to predict the properties of interest are just not at the same level as some of the toxicity models. So are you able to talk much about that?
I tried to give you an indication of what we're doing, and how. So at the moment, we probably have different steps on this. I think we are going to try to break down into the different test method types later on. We're really only mostly using the 301 and 310 OECD guidelines at this
Mm-hmm.
stage to do the first step of the model. But they can also include the extended versions, so we can go up to 60 days on the data. And we are looking at data that goes up 60 days. So we're hoping to include like things that go as far as saying, well, inherently biodegradable, by excluding things that would generally be considered as not persistent. We're not including at this stage things like the 307, 308, 309 might come later on, but I think we can already get a lot of feeling. I think ultimately, again, with SSBD, the way I look at it, it's if you go as far as having to do a 307, 308, 309, it's probably not really a very good sign for the sustainability of your substance. So let's try and pick out the key indicators of what can ultimately lead you there in terms of your structure beforehand. So ultimately, our model is going a little bit beyond the standard by incorporating AI in terms of machine learning, to be able to tease out the pathways of mineralization to a greater degree than is currently available. What this has meant is it's been a kind of shock that I've never had huge amount of faith in the 301s. I ran them myself when I was a study director, and I was lucky enough to have a really good microbiologist, Kees van Ginkel, in my group when I was in Axel Nobel. He's a really well-known
Yes.
microbiologist, and he's managed to teach me a few things. Um, I don't consider myself to be an expert in biodegradation, but enough to struggle by on the different study types. I've never really had a huge amount of faith in the 301 set, because there are so many uncertainties. Unlike ecotoxicity, you don't have all the precautions of the analysis to go with it. So you're entirely dependent on the technician getting the nominal concentration right in the bottle. You're looking at inocula, which come from a whole bunch of different wastewater treatment plants which are supposed to be predominantly municipal, but that doesn't mean exclusively, because of course there probably aren't any. But then it depends very much on what is the local effluent which is going in from industry into that municipal wastewater treatment plant. And certainly it would appear that that could lead to adaptation of microorganisms. There are also seasonal differences you can see that wastewater treatment plants don't function exactly identically if it's in wintertime or is in summertime or spring or autumn. There are differences in microbial communities, and then you have the difference between the studies. If you're using a 301D, the closed bottle test, your microbial diversity is probably going to be significantly compromised because you're going to have a much smaller bacterial community. So you need a competent degrader to be in your bottle. If you're using a 301B test, then you have a much bigger microbial community in there and microbial diversity. But at the same time, it's a kind of air flow through test. So you could be, if your substance is slightly volatile, you might be losing it, or it could be toxic potentially as well. And I really don't believe in the toxicity controls. They seem to be. I have a theory that possibly it's actually if in the toxicity control, those bugs which are not actually subject to the toxicity may be still very capable of degrading your reference substance. And all those bugs which actually would have been capable of degrading your test substance have possibly been wiped out by the initial mechanism of toxic action that occurs specifically to those bugs while they are attempting to degrade it. So what you can find sometimes is in your test substance group, you get a reduction in oxygen consumption, which is actually greater than you're finding more control in your blank. And that is something that Case taught me was the place to look to see if there was toxicity occurring. And quite often you'll see this in the 301 studies. If they put a graph in there which includes negative results, and you'll see it within the first 2 or 3 days. It'll drop lower than your control, and then it will maybe start increasing. But for me, that's where the toxicity needs to be looked at, not the toxicity control.
I've seen that in biodegradation data, and hearing that Kees van Kinkel's picked up on that as well is very, very good to, I think, capture.
It's so rare, isn't it, to see a toxicity control that says, "Oh, this substance is toxic." It hardly ever happens, and yet the cases where your test substance group drops below your control group, I see this quite frequently, and especially in substances that I might suspect have some toxic interactions. So all this it's a little bit aside, but what it's saying is it makes producing a QSAR for these kind of things, these kind of substances, very, very complex. You get differences within the 301 group. You get differences between maybe even the same method run in 2 places on the same substance, or even in the same place on the same substance if they do it at 6 months apart. As you've got no analytical backup really about it, except the occasional MITI test, which might have run some GC on the test substance to see whether it's there or not. And that's only an indication of primary degradation. So it doesn't give you a lot of detail. The fact that the MITI inoculum was also perhaps not the best when it was a mixture of 10 different places that was classically used in Japan, some of them coming from marine, some of them coming from soil, some coming from freshwater, doesn't help that study. I think it's pretty good in terms of very readily biodegradable and very persistent, but it's all the in-between ones which are so difficult to capture. So we started off with a data set. I can't remember how many thousands, 25,000 substances or something like that. It's whittled it down to 2,500 valid studies. And now, having taken those, we're finishing up on the clustering process. So we've clustered them by groups, structural similar groups. And once that was been finished, and we're really getting towards the end of that now, of looking study by study, sometimes again going back to the ECHA dossier, looking at the pertinence. Does it make sense? Does it not make sense? Seeing sometimes diversities of results which are astonishing. and trying to question with the chemists, does that make any sense to you at all, of why we are seeing one substance which is almost the same structural identity as a second substance, but one is persistent, less than 20% degradation, and the other one is readily biodegradable 80% within 15 days? Does this make any sense to you, or is there something seriously wrong with one of the studies? And I also believe that most of the time you're many more false negatives than there are false positives. It's actually quite difficult to get a ready biodegradation result. It's quite easy to get a result saying there isn't any ready biodegradation. If I see two substances, one which are almost identical, and one says it's ready and the other one says it's persistent, I tend to believe more in the ready one than the persistent one. But you can't be sure. We are trying to create sets now of clusters that we can be sure of, and then other groups where we're less sure, and we will try to let AI solve the mysteries of what is maybe explains the difference between those. And we will then come back to that, and we will look in detail to see if the mystery solving makes sense or not. And that result will then end up being included in our model by splitting. So you creating fingerprints, then we'll be splitting our models into three groups: the training set, the internal test set, and then the external validation set. And then from there, we will finalize our model. So I'm hoping that's going to come out really early next year. Then next steps, then I keep my fingers crossed on a research project. There's some interesting stuff going on in the world of MOX, more than one chemical substances. So in the world of MOX, there are some interesting issues around essential oils, and there's a bid by essential oil producers to try to demonstrate that their essential oil is not the same in terms of its properties and effects as the sum of its individual constituents, so that the classical recommendation for just kind of using additivity approaches is not appropriate in the case of essential oils. So I'm hoping that we will get a project next year which will be looking at multi-constituent substances. And then from there, the aim of that project, which is a 2-year project, would be to try to create an in silico model for multi-constituents, which would be a first. That depends a lot on whether there actually is some synergy or at least interaction between the individual constituents that could lead to a different outcome from if the substance are tested as monoconstituents. That's the basis of the hypothesis. We'll see whether it is true or not, as some substances within essential oils have not got a ready biodegradability label on them at this stage.
So this is for biodegradation. Very interesting. Oh, well, I wish you all the best in that bid for the research project. And thank you for talking me through some of the mechanisms of how that model's being put together and the methodology behind it. I hear a lot of what you're saying about the challenges with interpreting these tests. I would say that with biodegradation data, well, with all the data that we're dealing with is often, or most of it is biological, so there's an element of biological variability. But with biodegradation, there is a big, thick layer of biological variability, which can probably lead to quite some frustration when you're trying to uncover trends in data. But, you know, as you've mentioned, I think something that's good about your approach is that you are taking a close look at studies. You're not just taking data on face value. You are actually trying to understand what is sort of behind that data. I guess there's a challenge there in that, you know, you're not too kind of stringent in cleaning up data, because you can possibly throw out reasonably good data, you know, if you're not careful, and there's always a kind of risk of bias in that sort of area. What I would like to ask you is, are you mainly working with ECHA data for the development of these QSARs? And how are you dealing with ECHA data since they've moved to the ECHA platform? Are you grappling with that?
Yeah, so the answer is we are actually starting a Horizon project, which is working on pharmaceuticals. So we are moving to different areas. It's been a big challenge, as I said, keep it simple, stupid, and we start with the simple stuff. But I think we're actually getting to the point now where we are capable of looking at pesticides and looking at pharmaceuticals, and we're starting to do that in both of those cases, biocides ultimately. That being said, therefore, we do use different areas to pick up our data from. So, obviously, you don't have that much in the way of pharmaceuticals on the ECHA database, maybe a few, but there's not that much. But there's still a very strong emphasis in terms of the chemicals area, and certainly on the human health, we'll mostly be concentrating on that before we get carried away on pharmaceuticals. We're definitely not there yet. So yeah, ECHA disseminated databases is a major source for us. It's not an exclusive source, but it's probably 85% of the data that we look at. And despite what I've said about the validation of the data, sadly, the quality of the robust summaries is not always there either. The so-called Klimisch 4s, or sometimes they're Klimisch 2s, considered valid, but actually they're half empty based on almost nothing. Those are frustrating. To use for as a validation support. And typically what we do, we created a methodology also because of that frustration in-house, which we call the holistic approach, where we don't just use the specific study in question. We will go through all the relevant data, either for the human health people or on the ecotox, and sometimes we will even blend those two and start looking for information from on the other side of the fence where people may not normally go. But we'd be looking typically, it may be something has been put down as a Klimisch 3 example because there was no analytical method which was used and everything's based on nominal concentration. So yeah, I would totally agree that would be an unacceptable study most of the time. But then it turns out that right next door to that, you actually have the study was run on a fish in semi-static conditions where the water was changed every 24 hours. And then you have a Daphnia study where actually it was measured all through the study, and they did a static test and it lasted 48 hours and the substance remained completely identical. Is it then reasonable to throw out that fish study when you have, the only thing that could go wrong is that the technician really messed up in terms of the nominal concentrations? That doesn't usually happen in GLP lab. It seems like a reasonable read across. And then the question is, after that, does it all then make sense within the predictions that you're getting? So we've developed a method where if you have, let's say, a fish study, you could actually reverse engineer your calculation to go back from an algae study to determine what the water solubility would have been given that result, and then recalculate the fish study based on the water solubility. And you do that for each one. So you go through from log P, you go from algae, you go from Daphnia, and maybe even a chronic study occasionally, and you come back to the study that you're looking at, and you check each single result against the result that you have, and you can get an indication of whether it actually is a valid result or not as well. So there's a different approaches that we can use by combining the available data that might be on the disseminated database together with endpoint outcomes and the other robust summaries. So that's one reason why it takes us so long. I think of the 10 years that we've now been in existence, shortly, probably if we look at it, about 7 of them are spent just on data validation. So it is creating QSARs is not
it's not
quick, trivial, quick process. It's to produce ones that you really have confidence in. You have to be sure of the data. The good news is that once you start putting all these data points onto a graph, most of the time they make a lot of sense. You don't end up with a big potato on a graph, like if you just take your data set without checking it first.
And in terms of the ECHAChem portal, have you found that easier to work with?
Oh yeah, that was your other question. Yes, so no. Yeah. No, but I can't help wondering if they're not doing it deliberately because they're thick of people sucking out masses of data and making their servers crash every 10 minutes. I think there is clear. I mean, I see it in publications that are coming out. I mean, people are sucking data out of that database on a very regular basis without, and bioinformatics people to use it for AI, for example, and big data, machine learning and stuff, and so they can get a quick publication in. and probably Ecker's had enough of, I can only assume it's really for that, because in
Yeah.
terms of actually checking it, the still existing currently previous database is much easier to use, there's no doubt. Much easier
Yes.
to check.
I agree. To me, it feels like it's a step back. I know they've sort of done some steps to improve the functionality, but they have had some issues because I think there are around 1,000 dossiers or 1,000 substances that are still not on there. A lot of the interoperability of being able to search the database doesn't seem to be there like it was with the previous data set. Ultimately, to me, it feels like a bit of a backward step because we are moving into this big data world. And there's so much data in that database. There's so much for us to learn about
Yeah,
the fate and effects of chemicals
yeah,
by looking at the data on the whole. I think a lot of the new studies that are now being produced as a result of REACH dossier evaluation, where ECHA prescribes a certain test and insists that it must be written up in the dossier by a certain date, the quality of the recording of those studies should inevitably be better. So my hope is that in the future we'll have a better way of getting access to these data so we can really start learning something about chemicals.
yeah. I mean, we can denigrate it from that perspective, but I still have enormous admiration for that database. I think it is, for me, it's still the most useful database. And I admit I'm not a great expert on others, Kemble and various others like that, but I think that the typically a good robust summary really does give you as much what it's supposed to do. It really give you as much detail as you probably want from the study report itself, and in a specific format, so you know exactly where to go, and you can go through that in a tenth of the time it would take you to do the same thing if you had the study report in your hand. So if the robust summary is written well, I still think that there is an enormous number of cases, I see it every single time I open a dossier, where they should probably tell the people, the lead registrant, to go back and review their copy and upgrade the robust summary to include more information. Because that is, for me, still a major. They've accepted the dossiers, they've accepted the endpoints, but very often they could be written so much better. And some of them are just plain wrong. The conclusions are completely false if you actually read the content of the study, and
Yeah.
then you come to the conclusion. You go, hang on, that wasn't the conclusion. I mean, sometimes it's quite shocking. Somebody
Yes.
should go
Yeah. I
back
think.
and check them out.
Yeah. Definitely. Well, there was a lot of noise made around 5 or more years ago about the quality of the information that was in REACH dossiers, and I understand that there's been regulatory initiatives and also industry initiatives to improve the quality of the information in those dossiers. And I guess one of the challenges with this ECHACHEM database is that it's difficult to see whether those improvements are actually materializing. But you're absolutely right. The REACH database has been an absolute game changer for data availability on chemicals, and we should give EU
Credit.
credit
It's
for bringing that to the world. Yeah.
absolutely. I don't think we could have done what we've done within Kreatis if that database didn't exist.
Yeah. Yeah. Well, Paul, we have had a really great conversation here. There were lots of other things that I wanted to talk with you about, but I guess we'll probably have to have you back at some point to go into issues like UVCBs, dive into what you're doing at CTAC. I guess just a quick one on CTAC. So you're involved in the Vienna program.
Yeah,
How's that going?
it's going well. It's been, well, the members of the team, I'm deeply impressed by them. They've gone through every single abstract. I did a tiny part of the task I was allotted, was tiny compared to what many of the others have achieved, and there was not one abstract that went unturned, not even for a poster of any description. Every single poster abstract was read in detail. The committee had we had immense, interminable discussions sometimes on individual abstract, on whether there was enough information in there and whether we should send it back for a rewrite, or whether it. And it's I had no idea that this was going on at the level of the CTAC organization committee. For the people involved, the scientists involved in the CTAC organization committee, hats off. They're really, really dedicated. And I think they are just concerned that they want to make CTAC a really top quality congress, and certainly we spent a lot of time on it. And the next step, everything will go now to the chairs. There's been a lot of discussions on merging. There was so many. I think there were 2,500 different abstracts submitted.
Wow.
And then it's all a question of, like 40% of them were platform proposals, and you just could not have, all those co-chairs, and they will then have to make the decisions in terms of the oral presentations as opposed to which ones will get downgraded to posters. But all I can say is, it's been an eye-opener for me. The scientific integrity of the committee it's fantastic.
That's really great to hear. And are we on course for the biggest conference yet? Is it going to be bigger than Seville?
No, I think it'll be slightly smaller than Seville, but it's still going to be big. Of abstracts submitted and the number of attendees, really strong, year after year. And it'll probably therefore be a little bit smaller than Seville. It's still going to be a big one. It's going to be a good one.
Okay. Yeah. Well, the growth has been huge over the recent years anyway.
Yeah.
It had to tail off at some point. Great, Paul. Well, this has been fantastic. So what I do at the end is I just ask you 2 rapid fire questions, but we keep it really short because I know you need to run. So what's a goal that you're going to be working towards in the coming months, personal or professional?
Professional, it's and personal, I guess it's kind of the same thing in many respects. It's going to be long-term effects. So biodegradation on ecotoxicology, bioconcentration, that's a longer-term goal, and long-term effects on human health are really the areas we want to improve on.
Brilliant. And then the second one is, you start your career out today. What's one piece of advice you would give yourself? I don't put you on the spot there.
Yeah. Don't lose track of your original goal. You can be flexible, but don't lose faith in yourself. Just make sure that every time you fall over, which could be a lot, you get up again and you put it behind you and you just keep facing the direction you've been facing at the beginning.
Beautiful. Thank you so much, Paul. That's been fantastic. And I just want to say thanks to everybody who's been listening. Your time is really precious. So I'm so grateful that you've spent it with us. If you've enjoyed this podcast, please like it, comment on it, subscribe to it, and spread the word about chemical journeys. We've got more conversations like this to follow. But for now, I'll say thank you very much. And thanks a lot, Paul. And we'll see you next time.
Thank you for inviting me, Chris.